For our project, we tested which detergent works best for an at-home DNA extraction lab.
If we use liquid detergent, then we'll obtain more DNA from our strawberries because liquid detergent has a greater soap concentration, which dissolves the phospholipid bilayer of a cell more effectively. Materials include liquid and powder detergent, extraction buffer, 2 test tubes, a funnel, strawberries, milligram scale, a gram scale, rubbing alcohol, a pipette, and cheesecloth.
First, you must prepare the extraction buffer. You do this by using 3 simple household items: detergent, salt, and water. You are going to add all of these 3 ingredients into one beaker. You need 2 tablespoons of detergent, 1/2 teaspoon of salt, and 1/2 cup of water. First, you put 25 grams of strawberries into a plastic Ziploc bag. Then, after removing the air, you squish the bag for 1 to 2 minutes or until the strawberry is liquefied. Next, you add your extraction buffer into the bag with your strawberries. Next, you gently knead the mixture for 1 to 2 minutes. Try to create the least amount of bubbles as possible. Cut out a small 4-inch by 4-inch piece of cheesecloth and place it on top of the filter. Pour the mixture into the funnel. Do not let the edges of the cheesecloth fall in completely. The cheesecloth should filter out the larger pieces of strawberry. Next, you pour in the rubbing alcohol. You should almost instantly see DNA begin to form between the rubbing alcohol and your original mixture.
Remove the DNA with a skewer stick. Weigh the DNA by placing the skewer stick onto the milligram scale. Make sure you weigh the skewer first, then subtract the weight of the stick from your total weight. The amount of weight you have left is the weight of DNA.
Finally, you repeat all the steps with powder detergent. We conducted the experiment 3 times using liquid and powder detergent and pure soap to extract the DNA from each trial. As you see, we used 25 grams of strawberry each time we extracted the DNA. Throughout the first trial, we failed to extract any DNA using each detergent. After looking back on our notes, we discovered that we had incorrect measurements for our extraction buffer. We corrected them, remade our buffer, and continued with trial 2. For trial 2, we successfully extracted 1 703,000 milligrams using liquid detergent, 1 41,000 milligrams of DNA with powder detergent, and 2 139,000 milligrams of DNA using pure soap. In trial 3, we extracted 1 6,500 milligrams of DNA with liquid detergent, 1 81,000 milligrams with powder detergent, and 2 21,000 milligrams with pure soap. The average mass of extracted DNA for each detergent was 1 6,765 10,000ths of a milligram with liquid detergent, 1 61,000 milligrams with powder detergent, and 2 800 milligrams with pure soap. As expected, we extracted the most DNA using pure soap. Our hypothesis was correct, and between the liquid and powder detergent, we extracted the most DNA using liquid detergent because of its higher soap concentration.
Welcome to the Talk Show, starring Dr. Starkey Sova!
Hello, I'm Dr. Saki Sova, and today we'll be discussing the many contributions DNA extraction has made in society. As many of you know, the development of science is crucial to the advancement of society. In many ways, the discovery of DNA has contributed to the many benefits we have today. After all, it did spark the riveting Human Genome Project in 1990, which was completed in 2003. The purpose of this project was to identify and map the thousands of genes in the human DNA and determine the sequence of billions of chemical base pairs, altogether known as the human genome. This international collaboration has successfully collected and recorded almost all genetic contexts and data. Motivated by the Human Genome Project, many common complex diseases were discovered and raised into awareness. 3 years after the project was launched, breast cancer was confirmed to be associated to a number of genes. Soon after, Alzheimer's disease was added to the list, and 2 short years later, colon cancer was joined the family. From this project, scientists were able to grasp a better understanding of the ways to treat and prevent disorders that affect the human population. Furthermore, the understanding of awareness of the human genome may be vital for solutions to possible medical dilemmas in the future.
And that was Dr. Sarkisova. Thank you for tuning in.
Hello everyone and welcome to Terrific 2's Tedious Interviews! Today 2 will be interviewing the lovely Dr. Frederick Meinster, the scientist and founder of DNA extraction. Ladies and gentlemen, Mr. Frederick Meinster! Dance! Mr. Meister, welcome to our show!
Good morning, Mr. Frederick Meister. Please tell us about your latest discovery.
Good morning to you too. I most recently discovered how to isolate DNA while examining pus from a nearby infirmary that I took off of the bandages and stamen sperm cells.
Sperm cell.
Sperm cells.
How interesting.
Oh, no,
So, do you always wanted to be a scientist when you were younger?
Sorry, what?
Do you always wanted to become a scientist when you were younger?
Become a scientist?
Yes.
No. When I was younger, I wanted to be a priest. Yeah, you know. As a child, I wanted to be a priest, but my father would not let me. Oh, those were not the days.
Junior, come here, yeah?
Yeah, Dad!
Daddy, I want to be a priest, yeah? No, yeah? Yeah? No!
Oh no.
So, were you a popular kid when you were in school?
No. You see, as you can tell now, I have a hearing impediment. I cannot hear. Like right now, I don't know what the hell I'm saying. For all I know, I could be. But I know that's not the case because I can hear a little bit. But I had a hard time hearing. I couldn't really relate to the other students. It was hard.
Okay. Thank you for coming here and talk with us, Mr. Frederick Meister.
Say thank you.
Thank you.
You're welcome. Let me say that again. No, stop. What?
What?
Am I your guys' assistant?
Leave! Go away! You gotta go away. I'm so sorry. You gotta go away.
Oh my god.
I like worse in real life.
Bottle. It's really good.
Oh my god! Wait, oh! It's recording.
Good. Just say
No! You're done!
Hey,
Good looking.
Oh yeah. That's the stuff. Ow. I'm most recently. Okay. It goes faster, okay, Mr. Schneider? My name's Meinster. Meinster.
Go!
Meinster. Sperm cells!
Sperm cells!
Were you